Sir is an alumnus of our own college, and after that he pursued his MSc and PhD from Wayne State University, Detroit, USA, and later started company in Canada which worked on Alzheimer's disease. Sir had published more than 20 articles in international journals, and then now he started the Center for Advanced Applied Biological Sciences and Entrepreneurship in Rajahmundry in 2019. And already some of our students, UG students, are working with Sir, and they presented also. So introducing and welcoming Sir onto the stage to give away the talk. Thank you.
Uh,
Good afternoon, all. Thanks for inviting me here, and I'm really happy to be here. And I've been evaluating all your talks, and I have to admit, all your talks are so they're so good. So it's really a tough job as an evaluator for me to actually give you points. So you all did a great job. So today I'm going to talk about the latest trends in medical biotechnology. Specifically, so there are so many latest trends in medical biotechnology, but specifically today I'm going to focus on one topic. They're called the PROTACs. So if you heard the talk before me, some of my students, they talked about the HIV and they talked about the APOTAC and PROTAC, right? So what are these PROTACs? So I'm going to give you an overview of what is a PROTAC and how is that used in the medical biotechnology. So these PROTACs are small molecule drugs. They're like any other drug such as paracetamol or ibuprofen or anything like that. So the PROTACs have been highly used in oncology, in cancer biology, to treat cancers. So as you can imagine, the types of cancers that the speaker talked about this morning from GSL Medical College, there's so many types of cancers. So PROTACs were primarily designed to target different kinds of cancers. But today, I'm going to try to convince you that PROTACs can also be used for other kinds of diseases, such as the infectious diseases, and not just for the cancers. So before I explain what the PROTACs are and how you use these PROTACs in medical biotechnology, I'm going to have to explain a little bit background about how your cellular defense mechanism fights bacteria, viruses, and other pathogens. Because we take medications only when your body fails to tackle the infections such as bacteria and virus. But your body, every single cell in your body has its own defense mechanism. So human body is a highly advanced, complicated system. Okay? So we are the most highly evolved, complex living organisms on this planet. Human beings are the most complicated. Okay, so before we go into the details of the PROTAC, you all should know a little bit about the ubiquitin proteasome system. So the ubiquitin proteasome system is the primary mechanism that is present in most of your cells inside your body. And this ubiquitin proteasome system actually is a defense mechanism in your cells. The ubiquitin proteasome system actually fights bacteria, viruses, other pathogens such as fungi, and other pathogens. So, and the ubiquitin proteasome system is also a part of the defense mechanism in cancer biology. So this is your primary defense mechanism. This is not immunology. So please don't get confused, okay? Ubiquitin proteasome system has nothing to do with immunology. It does not make antibodies. There are no antigens. So this is a different kind of defense mechanism that every single cell in your body has. Okay, the abbreviation is UPS, the ubiquitin proteasome system. So what is this ubiquitin proteasome system? The ubiquitin is actually a small protein at the size of 8.6 kilodaltons. Okay, so it is a very small protein. This is the three-dimensional structure of ubiquitin protein. As you can see, there is one alpha helix and 4 beta strands. Okay, it is a very small protein. And ubiquitin, the addition of ubiquitin to any of your cellular proteins is called ubiquitination. So it is a post-translational modification. So you all know what a translation is, right? So you have genes in your nucleus, and you make it mRNA through transcription, and then the mRNA comes into the cytoplasm, and there is a translation to make the protein, right? So this is the basic dogma of the molecular biology. So ubiquitin is actually added to some of the proteins as a post-translational modification. And one important thing about this ubiquitin is it can form polyubiquitin chains, okay? So if one ubiquitin, when I say one ubiquitin, I'm talking about one molecule of ubiquitin. If one molecule of ubiquitin is attached to any of your proteins, that means it has a specific signal inside your cell for one of the cellular processes. But if there is a polyubiquitin chain that is attached to one of your proteins in the cell, that means that protein is marked or flagged for destruction, okay? So let's say you have a protein in your cytoplasm and you don't want that protein, okay? Let's say that protein is misbehaving. It is not doing its job. So your cell wants to get rid of that protein because it is misbehaving. So what it does is it uses the ubiquitin proteasome system by which the cell attaches a polyubiquitin chain to the protein that is misbehaving, okay? And when the protein gets attached with a polyubiquitin chain, that protein is chopped into pieces, okay? It is chopped into small pieces. And so during this process, the ubiquitin molecule is actually recycled. The ubiquitin molecule is not degraded, it is recycled. So you don't need a lot of ubiquitin molecules, okay? So once you target a protein to the proteasome, proteolysis, then you can recycle the polyubiquitin chain and then you can reuse them. All right, so ubiquitin is one of the players in the ubiquitin proteasome system. And the second player is the proteasome itself. So this is a complex of so many proteins. So this part has about 15 to 20 proteins. And this part, half of this part has 28 proteins. So you get the idea of the size of one proteasome molecule, okay? So on this slide, you have to, I'm going to take you through this mechanism, and you have to pay close attention, because if you do not understand this, you probably will not understand the rest of my presentation. Okay, so let's say you have a protein that is misbehaving in your cell, and you don't want that protein in your cytoplasm. You want to destroy that protein. What happens is there is an enzyme called a ubiquitin E1 enzyme. So to this enzyme, one molecule of ubiquitin is attached, okay? So the E1 ubiquitin complex, it exchanges the ubiquitin with a E2, so that ubiquitin that is attached to E1 gets transferred to E2. So instead of having ubiquitin E1, you have ubiquitin E2 complex. Okay, so that is the second step. The third step is the E2 ubiquitin goes and binds the E3 enzyme. So there are so many different types of E3 enzymes, and I'm showing you only two, the ring type and the hect type. But in reality, there are so many different kinds of E3 ligases, okay? So the E2 ubiquitin comes to E3, and then the ubiquitin that is attached to E2 gets transferred to the protein that you want to destroy. Remember, this is the protein that you want to destroy because it is misbehaving, right? So the ubiquitin molecule started its journey from here. It got attached to E1, then got transferred to E2, and then got transferred to the final target protein by E3. So your unwanted protein eventually builds up a ubiquitin chain. So this is the ubiquitin chain. Once a protein has a polyubiquitin chain, that is a signal for the proteasome. So this is proteasome. So that is a signal for the proteasome to catch that protein and cut it into pieces. Okay? So once the ubiquitin is transferred onto your target, you have a polyubiquitin chain, And this polyubiquitin chain is detected by the proteasome, and it chops your protein into pieces. So that's how the cells get rid of bacteria, viruses, and other unwanted proteins. Remember, this has nothing to do with immunology. No antibodies, no antigens. This is purely a cellular defense mechanism. All right, so remember I told you there is the proteasome, right? So now I'm going to zoom into this part, the whole of this blue color part, and then half of this CP. How does it look? This is how it looks like. So remember, this is the red one is the protein that is misbehaving. So you don't want that protein in your cytoplasm. So what did you do? You added a polyubiquitin chain. So there are 4 ubiquitins that are added to this. By the way, this whole diagram is actual molecular modeling. So it is actually real. It's not predictions. Okay, this is how it looks like at molecular level or atomic level. So these 4 ubiquitins are detected by this RPN10. As you can see, the ubiquitin is detected by the RPN10. And this last ubiquitin is detected by the RPN13. Okay? And that's how the ubiquitin, polyubiquitin tail is docked onto the proteasome in such a way that the unwanted protein that you want to destroy comes into the active site. Once your protein comes into the active site, the polyubiquitin tail is cut off. And once the polyubiquitin tail is cut off, these ubiquitin molecules are recycled. They're used again in the next cycle. And the protein that you want to get rid of is unfolded. Okay? Proteins are usually folded. Now the proteasome, it unfolds your protein, and it slowly passes this protein sequence through the catalytic particle of this proteasome, so that this protein chain goes through here and gets chopped up into pieces. Right? So if this is a bacterial protein, you just kill the bacterial protein. Or if it's a viral protein, you just kill the viral protein. That's how your cellular defense mechanisms work. Now, I want to explain what is the PROTAC strategy. PROTAC is a new technology that has been widely used in oncology or cancer drug discovery. So the PROTAC strategy is basically based on the ubiquitin proteasome system that I just explained to you. What happens is, in the case of a PROTAC molecule, you have part A and part B. So part A, it's highly specific to the protein that you want to destroy. Okay? For example, let's take cancer. In cancer, one of the proteins that is always misbehaved in most of the cancers is RAS, R-A-S. RAS is one of the proteins that always misbehaves in most of the cancers. So let's say you want to remove that RAS from the cytoplasm of the cell, right? So you design the PROTAC in such a way that this black part, which is part A, binds to RAS. Okay? So this is your target protein that you want to destroy. You don't want this target protein because it is misbehaving. So you design the molecule in such a way that this part binds to the target protein that you want to get rid of, and the other part binds to the E3 ligase. Remember, E3 ligase is the final step in the ubiquitin proteasome system, right? So this is the E3 ligase, which transfers the ubiquitin molecule onto your target protein, right? So similarly, in PROTAC, you have part B that binds to E3 ligase, which actually brings in ubiquitin molecule, and this ubiquitin molecule is transferred onto the target protein. Eventually, the target protein gets a polyubiquitin tail, like this. And when the target protein gets the polyubiquitin tail, then what happens? It gets degraded into pieces by the proteasome. Okay? So that's how we get rid of this misbehaving protein in cancer or AIDS or malaria or any disease. So whatever disease that you want to work on, you see that protein that is misbehaving, and then you design the drug using the PROTAC strategy, in which this part binds to the protein that you want to destroy, and this part binds to the E3 ligase, so that the E3 ligase puts on ubiquitins, and you have a polyubiquitin tail that destroys the protein through proteasome. So conventionally in drug discovery, what we do is you have a drug target and you design a drug. This drug binds to the drug target, okay? But you know, there are different kinds of interactions between molecules. You have covalent bonds, ionic bonds, and van der Waals interactions, right? So if you have covalent bonds, that means the two molecules are permanently bonded together. You cannot release them. They're formed into one single molecule. If you have ionic bonds or van der Waals or hydrogen bonds, then these two molecules will bind, but they can be released. Okay, so it's not permanent. So in this case, in the normal conventional way, we design the compounds, the drugs that bind to the protein, but they come off because they're not forming covalent bonds. They're only binding the protein with van der Waals and ionic interactions. So they bind and they come off. Okay? So when the drug comes off, then the protein will misbehave again. Right? So, but in the PROTAC strategy, what happens is you take the misbehaving protein and you have a PROTAC that brings in E3, and your misbehaving protein is completely degraded permanently. That misbehaving protein is no more, right? It is destroyed. So that is the difference between the regular way of drug discovery versus using the PROTAC strategy. Examples of PROTACs. I remember in the morning somebody asked about how you make the PROTAC. So how you make the PROTAC is you have these molecules that are pre-made that are specifically targeting the misbehaved proteins. And these are the molecules that bind to the E3 ligase. So you can pick and choose any of these, and you can pick and choose any of these, and then you combine them with a linker. So that is a PROTAC molecule. That's how a PROTAC molecule looks like. So for example, how does it bind to the protein? So this is how it binds to the protein. For example, this is the E3 ligase, the red color. And the green color is the misbehaved protein. And this yellow is the PROTAC. So what the PROTAC is doing is it's bringing the E3 ligase to the vicinity so that it's getting added by the ubiquitins. And once you add the ubiquitin, then you know the proteasome will destroy that protein. Examples of PROTACs. So thalidomide is commonly used as the E3 ligand. So thalidomide has been shown repeatedly by a lot of scientists that it binds to the E3 ligase. And here you can actually have different kinds depending on your protein. And once you have the ubiquitin, it goes to the proteasome. Okay, so I don't want to take too much time. Just want to summarize one more time. PROTAC is a clean technology because when it binds to the unwanted protein, when it binds to the misbehaved protein, it will destroy that protein, right? But the normal drug discovery, when you design a drug, it goes and binds to the protein, but then it comes off, right? And then the protein will misbehave again. In this case, when the PROTAC binds to your misbehaving protein, it is chopped into pieces permanently. And so, like I said, PROTAC is a two-part drug molecule. It has a part A, part B. Part A binds to your misbehaving protein. Part B binds to the E3 ligase. And these two parts are linked by a linker. And the proteasome finally degrades your unwanted protein, the misbehaving protein. And one of the biggest challenges in the field of drug discovery is bioavailability. So for example, let's say you have a peptide, and if you take that peptide as a medication, what happens? Your stomach has pepsin. Pepsin cuts the peptide bonds, right? The peptide that you take gets chopped off into pieces in your stomach. So you have to design drugs in such a way that they do not have peptide bonds. And PROTACs, they don't have peptide bonds, but so they're mostly non-peptidic small molecules, which means they don't have peptide bonds. Okay, that's all I have. Thanks for listening. I'll take any questions. Questions? No questions means you understood everything, right? Or you didn't understand anything. Did you understand everything? Because I'll have to ask you questions. What should we do?
Sir, you said that it's like paracetamol or something like that, but it's a drug, right? So it should be taken by any time. If we get fever, we take the paracetamol. So whenever we want, we can take that, or like it should be a period of time something.
So the PROTAC, I hope you guys understood the question. The question was, you take paracetamol when you have a fever, right? Do you take the PROTAC similarly? The answer is no, because PROTAC is designed for a lot of different reasons. It could be designed for cancer, could be designed for HIV or malaria or any other disease. So depending on what symptoms you have, what disease you have to take that PROTAC only. Yep. More questions?
How does the PROTAC understood that that is the target protein, sir?
So how does the PROTAC reach the target protein? How does it identify? So like I showed on one of my slides, proteins have specific shapes, right? I showed you like a half